I'm going to be presenting my project, and it's on cancer. So this first slide is just a lung cancer cell during cell division. So just an overview, cancer is a disease that is caused by the accumulation of mutations and uncontrolled division of abnormal cells in the body, and mutations in genes that normally control and regulate cell growth and division lead to cancer. So genes that code for like growth factors, growth factor receptors, things involved in cell signaling pathways. And then it is also a multi-step model, so that just means that the longer we live, the more likely we are to develop cancer, because if cancer results from an accumulation of mutations, and if mutations occur throughout our lifetimes, then the longer we live, the more likely we are to accumulate a lot of mutations and therefore develop cancer. So that's basically what this says. You have a normal cell, some sort of genetic change, it becomes a cancer cell which divides uncontrollably, and then you end up with a tumor. Cancer causes, things like environmental influences can have an impact, so influences like from treatments, radiation from x-rays, or too much UV radiation from the sun. Also chemical carcinogens like cigarette smoke or certain viruses can also cause cancer. And then also genes. So proto-oncogenes are genes whose protein products stimulate cell growth, and so when a proto-oncogene is mutated, it becomes an oncogene. And if a proto-oncogene has products that stimulate cell growth, then an oncogene is going to have products that overstimulate cell growth, and that just results in excessive and uncontrolled cell division. Proto-oncogenes become oncogenes by three main causes: movement of DNA within the genome, amplification of a proto-oncogene, or a point mutation. And so this is just a little diagram. You have some sort of cancer-causing agent. It mutates the DNA in a proto-oncogene, which then becomes an activated oncogene, which promotes too much cell division. Then you have a cancer cell. Proto-oncogenes becoming oncogenes. So the first one is movement of DNA within the genome, which refers to translocation. So as you can see here, it's just the switching, like chromosome fragments will break and then rejoin and switch incorrectly. So you can see it's uneven here. And that can be a problem because it can cause a proto-oncogene to be translocated next to an active promoter region, which results in overtranscription of a proto-oncogene because it's going to be too actively promoted. And then its protein products can overstimulate cell growth or division, which turns the proto-oncogene into an oncogene, and that can cause tumor development. And then amplification of a proto-oncogene just results in the overproliferation of a proto-oncogene's protein products, and that can cause excessive cell growth as well, which can result in a tumor, Point mutations either occur in the proto-oncogene itself or in a control element which regulates gene expression. And so that can change the protein product to one that is overactive or resistant to degradation more than it normally would be, which is a problem because then you get a protein product that won't be degraded, so it'll just keep promoting cell division. And this is just an overview. Sorry, my head's a little bit in the way here. It's just an overview of all of these three conversions. Tumor suppressor genes are also an important gene in cancer development. These are genes whose protein products help prevent uncontrolled cell division or division, so they're like the brakes of a car because they stop runaway cell division. So when you press down on the brakes of a car, you're going to stop or slow down. So you're going to stop and slow down cell division. You're going to regulate it, make sure it's regularly occurring correctly. But when you take the brakes out, so when you get a mutation in a tumor suppressor gene, then you don't stop and you just keep on dividing. If you were a cell, you would just keep on dividing and end up as a tumor. And the mutations that prevent controlled cell division, and the tumor suppressor gene products also repair DNA, control adhesion or anchorage, which has a role in the spreading of cancer or cell cycle control. An important tumor suppressor gene is p53. So p53 encodes for a transcription factor that promotes the synthesis of the cell cycling inhibiting proteins, which are just proteins that stop the cell cycle. And so if p53 is inactivated or altered or some way dysfunctional, then the cell cycle doesn't have anything to regulate it. p53 can also activate other cells directly involved in DNA repair. So one of the genes is p21, whose protein product stops the cell cycle by binding to cyclin-dependent kinases. And then this allows the cell time to repair the damages or DNA mutations that happen, so you don't pass on those mutations when you divide. And p53 can also activate suicide genes if the DNA mutations in a cell are too great or too much, that they just can't be repaired, and so the only thing that a cell can do is go into programmed cell death, which is apoptosis. And it's called the guardian angel of the genome because it kind of watches over everything. It controls the activation of other genes that contribute to the cell cycle. It promotes cell growth itself. It's just an all-around great guy. And then this is just a little diagram. So this is a normal p53 protein, which when the DNA is mutated, it amplifies, which then tells the cell to go into cell suicide. But a non-functional protein doesn't do that, and it just sends no signal at all, and the cell is allowed to divide uncontrollably, which can lead to cancer. Apoptosis, as I mentioned earlier, is programmed cell death, and this is what happens when p53 tells those cells to go into cell suicide, which is apoptosis. So basically, the cell receives some sort of signal from outside or inside that tells it it's time to go, buddy. And these little enzymes called caspases are activated, which break down the organelles and membranes and the nucleus and DNA in a cell. And then it also tells the membrane to start to bleb, which is just like little protrusions on the membrane. And then it makes these little apoptotic bodies which hold everything from the cell in it, everything broken down. And then those fragments are digested by phagocytic immune cells, which are just like macrophages, which make sure that there's no inflammation and that there is also the caspases don't get out and start to digest the neighboring cells that don't need to die. And this just prevents damaged DNA replication or passing on the mutations, because that can also result in cancer, and cancer cells are often resistant to apoptosis. Angiogenesis is the formation of new blood vessels, which are capillaries, and they keep the tumor alive because they bring in and out nutrients and waste and oxygen to the cell, and that keeps the tumor alive. The cancer cells in the tumor can also creep along those capillaries, which is why cancer can spread, which is metastasis, the spreading of cancer. This is a specific example of cancer, glioblastoma malignant astrocytic glioma. It's a mouthful, we're just going to call it GBM for short. It's the most common and aggressive type of adult brain tumor, and its like trademarks are vigorous angiogenesis, and so like metastasis, which you can see here, it's spreading a lot. It is also apoptosis resistant, so treatments that trigger apoptosis don't really have an effect on it, and it is mostly contributed to by the RB pathway and p53 pathway. And so the p53 pathway in GBM, p53 is stabilized by the p14ARF gene, which basically, so in GBM, p14 is no longer expressed, and so there's nothing stabilizing p53, and if p53 is less stable, then it can't regulate the cell cycle correctly, which is bad. It can cause excessive cell growth, just a tumor, you know. And then it is also degraded by MDM2, and so MDM2 is amplified in cancer, or in GBM specifically, and it allows the cell to escape the regulation of the cell cycle from p53. And if p53 is being degraded, it can't regulate because it's being degraded. It's not going to be there anymore. So when you amplify MDM2, it's not going to be able to persist because it's going to be degraded. And then this just leads to unregulated cell growth. P14, loss of expression of p14 is contributing to about 76% of GBMs, and amplification of MDM2 only exists or is present in about 10% of GBMs. So the p14 gene, the loss of expression, has a much bigger role than MDM2 in GBMs. I hope that made sense. And the RB pathway in GBM is controlled by the PRB gene, which is a tumor suppressor gene. It inhibits the cell cycle, and so the PRB gene is inhibited by a CDK, and since it can't control the cell cycle anymore, you're going to get an overexpression or over cell division, which leads to cancer. Conclusion, super quickly, cancer results from the mutations and abnormal regulation of the cell cycle, so like the multi-step model of cancer development, which I have a diagram here. So you just get a mutation, a loss of a tumor suppressor gene, you maybe get a small growth, just benign, and then you lose more genes, something becomes an oncogene, you get a larger growth, and then you just accumulate more and more mutations as your life goes on, which can result in a malignant tumor. And GBM results in part from mutations involving the p53 and RB pathways. And then this is my bibliography. Thanks for watching. I hope you learned something. Bye.
That's all, folks.